Let's talk some theory. So this week you're conducting some staining, you are doing some microscopy, and you are interpreting results from your samples. And so things you need to understand and be able to use to interpret are cell morphology and arrangement. You need to understand when you're doing a gram stain or a simple stain what actually is going on. You need to know how a microscope works. You all have microscopes, and so when you're using it, you should have just some fundamental concepts of how it's actually working. We will talk about how we take information from gram staining and cell morphology, as well as other biochemical and physiological tests of bacteria, as a way to identify the bacterium. So this is important in clinical laboratory science to be able to identify, right, the infectious agent that is an infection. So let's go ahead and take a look at these slides. So this is just kind of an overview of looking at cell morphology, a diagram of a cross-section of a bacterial cell, gram-positive. You've already seen this before. And what happens during staining procedures to end up with an image like this. So these three images on the right are from the hands-on lab laboratory. So if you've not seen these yet before, you will when you're doing the exploration. And over here, let's look at these first. So bacteria are classified how they look under the microscope, their shapes. So a coccus is a sphere. Okay, so that's what it is. It looks like a little ball. And so over here in the blue and orange image, not the kind of microscopy that you will do, but you'll see that these look like a whole bunch of little balls. Okay, bacillus is longer than it is wide, and it's like a rod. And this is a bacillus in the upper hand right. So this is what it looks like under the microscope for the same kind of technique you'll be doing. Coccobacillus is in between. It's still longer than it is short, than it is wide, excuse me, but it's kind of intermediate between coccus and bacillus. Now these are the three main ones that I would test you on if I were to test you. However, in the lab you'll also be looking at a spirillum, really close to a spirochete. Don't worry about the difference of this for the purpose of this class. But they look like this, little corkscrews. Okay, this is what some Vibrio look like. So they named it Vibrio, and that's a type of bacteria that me and my students work on here at JU. And there's some bacteria that are pretty crazy that just have kind of weird different shapes, so don't worry about that. So that was cell morphology. Cell arrangement is, well, the arrangement of the cells. So all over here are the coccus, and so you can see in each one of these, there's a sphere, there's a bunch of spheres, there's spheres grouped together, there's clusters of spheres. And so instead of saying spheres, we say coccus or cocci. Cocci is the plural. And so diplo is the arrangement, cocci is the morphology. So you put it together, diplococcus would be two cells next to each other, two cells next to each other. And if you have more than one, so this is a diplococcus, this is a diplococcus, and together they're diplococci, it's just plural. So if you ever see a chain, that is strepto, okay? So this would be streptococcus. This chain over here is formed of rods, in other words, bacilli, so that's streptobacilli. And so down here we have staphylococci, which is what staphylococcus is named of. And when you look at Staphylococcus epidermidis under your microscope, you should see something that looks like this. So staphylo is this kind of bunched grape-looking arrangement, and it is cocci because they are made of coccus. So understand that this is arrangement we're talking about when we're talking about diplo, strepto, tetrad, staphylo, and be able to look at an image and be able to say what it is. So if I were to give you an image on this and ask you what is the cell morphology and arrangement, you would say streptobacilli. Now, fixing a sample essentially kills the sample. And when I say sample, I mean some type of microbial sample, either bacteria or fungi typically. Okay? And the fixation actually fixes them in position and preserves structures. Okay? So in lab, you carry out heat fixation, right? And don't worry about chemical fixation for the purpose of this class. And so you'll recall that you put a sample, multiple samples on slides, you let it air dry, and then you went 1, 2, 3 through the flame, which then fixed the sample onto the slide. So then the next step is to stain your sample. And the reason you do this is to increase the contrast. And by increasing the contrast, you are then increasing the resolution, which is the ability to differentiate between two objects. Okay. So during the staining, typically, well, the most common stains are basic stains, which are positively charged, and they're all dyes. So they're all colorful. And so positive, we know, is attracted to negative. And so in this instance, our positively charged dyes will bind to the negatively charged cell membrane of the bacterial cell. So here are three common simple stains: methylene blue, crystal violet, safranin. You'll see that the molecule has a positively charge associated with each one of them, as we just discussed. Methylene blue and crystal violet are blue. Safranin is pink or red. This is a sample that is not stained. This is a sample that is stained. Each one of these is an individual bacterial cell. And so you can see that this is increased contrast, and that is that your specimen is much darker than the background. So again, simple stain can be any of these. This is methylene blue. You will also be conducting a differential stain. Don't worry about these others yet. We'll talk about them later. And that is where you will use crystal violet, which is purple or blue, and safranin, which is pink or red. So a direct stain, which you're doing in lab, means that basically you just add one stain and it directly binds to the bacterium by binding to the negative cell membrane, negatively charged. And so in this sample, you see that the individual cells are purple. Okay, and that is because you used, for example, crystal violet. In lab, you're not conducting negative stain. Don't worry about that. And this image is from hands-on lab as well. So you should be seeing this when you're doing the lab exploration section. Okay, so let's get back to this differential stain. And so this image right here is also from hands-on lab. And what I did was I added this down here in the lower left to show you what the samples would look like before staining. Okay, and what this is showing you is you've got a sample of gram-negative. And so what would this cell morphology be? Would this be coccus, bacillus, or coccobacillus? That's right, that would be bacillus, both of these. Okay, so in this case, this is gram-positive and this is gram-negative. First step, you add crystal violet. It floods them, and it's a direct stain, positively charged, and it will stain all of the cells purple. So you rinse it off after that, and then you add iodine. Iodine will bind to the crystal violet and create big complexes, and typically your sample gets darker purple. I would like to see these a darker color in this image, but that's okay. Okay, and watching the video of me doing the lab also shows you more information on this. And then you rinse it with water, and then you use the decolorizer. And the decolorizer is going to decolorize what? The gram-positive or the gram-negative cells? That's right, the gram-negative cells. So at the end of this step, after you rinse it with water, what you'll see is that your gram-positive cells are still purple, and your gram-negative have no color. Now, under the microscope, it will be hard to see these gram-negative cells, so what you do is you use a counter stain. In this case, for gram stain, the counter stain is the safranin. Safranin, it can also be used as a direct stain, so it will still bind to both gram-positive and gram-negative, but you've already loaded up your gram-positive cell with purple dye, the crystal violet, and so even though this may get darker, it'll look maybe just darker purple. It may have a little reddish hue, but it'll still look very purple. Whereas the gram-negative cells are now red or pink. Okay, so that's how you differentiate gram-positive from gram-negative. Okay, so this is a differential stain. Up here, this is showing an image of a mixed culture with two different types of bacteria. So let's talk about the pink ones. These are what? Gram-negative, and these are rods. So this would be a gram-negative bacillus. So all of those. And then the purple balls, right, the spheres would be gram-positive what? That would be gram-positive coccus. And so this is nice because it shows you two different cell shapes as well as the gram reaction. So now let's take a look at what we think is actually going on the cellular level. So what we have here is the diagrams that you've seen before. On the left-hand side is cross-section of a gram-negative bacterial cell wall and gram-positive. So let's look at the gram-negative again. So gram-negative, we've got inside of the cell, down below. Then we've got the cell membrane, also called the plasma membrane. We then have a thin layer of peptidoglycan, and on the outside is the outer membrane with a variety of proteins in it. The gram-positive bacterium also, of course, has a cytoplasm and a cell membrane. It also has peptidoglycan, but it's a lot thicker. Okay, so we have multiple layers of thickness of peptidoglycan, and what is absent up here? That's right, what is absent of here is the outer membrane, which we have here in the gram-negative bacteria. So why does that matter? Why are we talking about that right now? Well, think of, let's get a couple ideas in our mind first. One is that everything, both stains, the crystal violet and the safranin, as well as the decolorizer and the iodine, need to penetrate to an area that will then hold on to the dye, or it will not hold on to the dye. So different structures and how they're built will result in different abilities to retain the dye during decolorization. That's key. Decolorizer can be alcohol, it can be a mix of alcohol and acetone, and one thing we believe the decolorizer does is it helps dissolve the outer membrane of the bacterial cell. So when we're talking about outer membrane, obviously what we're talking about is gram-negative bacteria. Okay, and the fact that when we add decolorize, it also we believe it dehydrates the cell a little bit, and so what it does is it shrinks things. Okay, so let's go ahead and then look at the image again, and imagine now what we've got is crystal violet flooding into the cell, coming through the outer membrane, going into the peptidoglycan and binding to this region down here. Let's just kind of keep it simple. It's going to bind to the cell membrane and within the peptidoglycan. Okay, it's going to be everywhere, okay, all up here, but let's focus on this area. So over here, we're also going to have the crystal violet binding to negative structures down here on the cell wall. And so crystal violet then fills up this area as well. When we wash it, any extra that's up above goes away. We then add iodine, and the iodine also goes into the same places as the crystal violet. And what we believe happens is that molecules of the crystal violet will bind to molecules of the iodine, and the iodine will kind of go all around it and make this larger complex. So in other words, crystal violet goes in, iodine goes in, and they complex, and now they're bigger than they were before. They're bigger than each one. And so this makes it harder to leave. Okay, so if we look at the diagram again, now what we've got is we've got crystal violet iodine complexes all in the area of the peptidoglycan and the cell wall. The peptidoglycan and the cell membrane. Okay, and then you add decolorizer. Decolorizer, we believe, will start to dissolve some of this outer membrane. Okay, so if this starts getting dissolved and we've now got holes in here, the crystal violet iodine complex can then leave very much more readily. Whereas at the gram-positive cell, there's no outer membrane to release. So then the question is, well, why can't it leave even more easily? If the outer membrane, right, if the outer membrane was a barrier for it coming out, well, since a gram-positive bacterium does not have an outer membrane, then it should be even easier for the crystal violet iodine complex to leave, right? However, what does the gram-positive cell have extra that the gram-negative cell does not, right? That's right, we've got the thick cell wall. So now think about a bunch of crystal violet iodine complex all down here, and then when we add the decolorizer, it kind of shrinks the cell a little bit from dehydrating it, and the mesh, if we think of peptidoglycan as a mesh like this, right, where normally the crystal violet iodine complex would be able to go through, instead, when you add the decolorizer, it physically kind of locks into place a little bit more. So now there's not as much space here. It's not like before where the crystal violet iodine complex may be able to leave quite readily, but now it's kind of tighter even more, and so it's harder for the crystal violet iodine complex to leave the gram-positive cell. And so it then leaves not nearly as much, so more is retained. Okay, so then you add water, as you do in the gram stain after every step, and then you add safranin, and safranin will go ahead and go through the peptidoglycan on both types of cells and stain them red. It's just, again, that the gram-positive cell also has a bunch of purple, and if you add red on that, it just makes it more darker purple or kind of a reddish purple, whereas the gram-negative cell, right? If we look over here, the decolorization made all of the crystal violet iodine complex leave. So now when you add safranin, it loads up this area with safranin to give you a nice red or a nice pink coloring. And so that's what we believe is going on, at least to some degree, during the Gram stain, and why gram-positive bacteria are stained blue or purple, and gram-negative bacteria are stained red or pink. And another very important concept is, well, why is it that water, you can rinse with water for a long time and the dye stays in, whereas when you start using decolorizer, it comes out? Well, that's because the decolorizer are organic solvents, and organic solvents dissolve the dyes a lot more readily than water does, in addition to disrupting the cell membrane and such. So that's one of the main reasons. So when you are carrying out the technique in lab, if you over decolorize, it will get to the point where even gram-positive cells can become completely colorless. And then if you stained it with safranin, it would look just like a gram-negative, because too much decolorizer, in other words, for a longer period of time that is recommended in the lab guide and the lab video, basically just enough time, and that organic solvent will keep bringing out the crystal violet and the iodine complex from it, so it'll keep dissolving it. So it's a balancing act in terms of time, and that's why in the lab video I talk a lot about making sure that you adhere to the time constraints. Okay, and down below, also from your lab, hands-on lab, is showing you, and you should see this as well. I talk about this in the lab video as well, and this is showing stained cheek and with gram-positive bacteria stained, and your cell does not have a cell wall, right? It does not have a peptidoglycan, and so your cheek cells should not retain the crystal violet iodine complex, because you don't have the peptidoglycan wall to keep that crystal violet complex inside. And so that's what you need to know for understanding how gram-positive and gram-negative bacteria, their cell structures, contribute to the final coloring that we see. A little bit of theory about what we think is going on.